Okay, everybody, we're going to go ahead and start for today's first online lecture. I've made this relatively brief. That way we can hopefully all transition to this a little bit more easily than a lengthier lecture. So we're going to get back into Chapter 6, looking at drift and selection, focusing on the the sections that I've got highlighted here. We'll also be revisiting the spotted gar case study with a different perspective, now thinking a little bit more about drift and selection and landscape genetics. So keep in mind the terms that are going to be on the next couple slides as we kind of revisit that last genetics section of our spotted gar case study. And then I'll talk a little bit about the guidelines for case study number 2, which again is going to be due this Friday at 11:59 p.m. and that will be due via Moodle. So make sure that you're turning in the material online through Moodle in a timely fashion there. Okay, so this slide should be pretty familiar to you guys by now. Looking at these major terms that we've talked about for Chapter 6. So again, you should make sure that you're familiar with these. Don't worry about this Word document information on the right-hand side. Again, the key information that you should be knowing are going to be these terms and concepts, and that's what we've spent time on over the past week or so. And these are some of the terms that I'm referring to in the case study 2 assignment for what you should be finding between the journal article that you've selected and also the journal article that I'm going to be assigning to you. Alright, so before we get into revisiting the spotted gar case study, I want you guys to look at case study number 2 as far as the guidelines and the specifications here. So first of all, you should follow the same guidelines you did for case study number 1. And so make sure you're following that same format, including the file naming system. Make sure that you've got that so that way we can keep everything organized. So what I want you to do for case study number 2 is I want you to identify and define the following terms from this case study and Chapter 6. So the article you're going to be reviewing for case study 2 that you're assigned is the David and Wright 2017 paper. So this is based on part of our spotted gar case study, so some of that should be pretty familiar to you, looking at mitochondrial DNA and some of the landscape genetics and genetic diversity of the spotted gar population. So I want you to take a look at these terms and make sure that you're identifying them from this case study, and that's going to go in your sort of write-up or summary of the case study, or it can go towards the end depending on where it fits. I also want you to compare the article that I'm assigning to an article of your choice, and this is going to be the journal article that you guys selected about a week ago and turned in on the note cards. Again, if you don't know what that journal article is, please make sure to email me. That way we can make sure that you're on the same page for the journal article. Within that journal article for the comparison, I'd like you to compare 4 of the above terms that you have here, either with the David and Wright assigned 2017 paper, or you can compare it back to the book with those particular sections of the book. So however you make your comparisons, again, similar to case study 1 with this. So you're going to be comparing an assigned paper, which is the David and Wright 2017 paper, to a paper of your choice, which is the one that you selected and put on the note cards about a week ago. Another thing that I want you to make sure that you're including on this assignment is to look at what's the deal with Texas. So I want you to think about, in David and Wright 2017, why is Texas different? So what are some of the possible explanations as to why that Texas population was different from some of the other populations we compared to? So again, if you've got any questions about this, please make sure to email me, and we can address those as they come up. So remember that deadline is March 20th. That's this Friday at 11:59 p.m. Okay, so moving forward, we're going to revisit the spotted gar case study, and again, you can follow this link to get to that case study, to get to the PDF of the article there, and we're going to be looking at genetic variation and biogeography of the spotted gar from core and peripheral populations. So again, a lot of this is going to be reviewed to you, but I want us to get on the same page going into case study 2, and of course, hopefully make our online transition a little bit easier. So looking at our overview from our spotted gar case study, we're looking at genetic diversity and biogeography. And so just to get back in gear with that, thinking about peripheral versus core populations. So by now this should be pretty familiar to you, but peripheral populations, when we're thinking about the spotted gar, is this sort of disjunct population up in the Great Lakes, and our core population is centered on the Mississippi River Basin and also some of the Gulf Coast regions as well. A couple other things to keep in mind are that the peripheral population is a much younger population. It's only about 8,000 years old, and it's a much smaller population. So if you remember our concepts of selection and drift, remember that genetic drift is going to have a stronger influence on small populations, and natural selection is going to have a stronger influence on large populations. So with those things in mind, we would expect that genetic drift is going to have a larger impact on the peripheral population, and natural selection is probably going to have a larger impact on the core population. That's not to say that we don't see natural selection or genetic drift working on both of these populations. It's just depending on which one might have a stronger effect or stronger influence based on population size. So, our objectives for this particular study and our hypothesis are as follows. We wanted to identify and explore genotypic relationships using mitochondrial DNA, comparing the core and peripheral populations. So, is there a difference in genetic diversity? And we're using something called phylogeography, which is looking at the distribution of genealogies. So, we're looking at sort of the geography of genetic diversity in order to interpret those relationships among populations. Remember that from our other aspects of this case study, we looked at growth of young of the year fish to look at if they grew faster and larger. We also looked at growth and reproduction and age of the adult populations, and we found that they were actually different too. So we've already seen that phenotypically speaking, these populations are different. We wanted to see if there's any genetic evidence that would back up those populations being different. So our hypothesis was that the genetic structure, remember genetic structure, population structure is something that we talk about in Chapter 6. We think that the population structure or genetic diversity is going to be reflected in the geographic position or location of core and peripheral populations. And the picture down here is some of us who basically got some of the samples as we went from Michigan all the way down to Thibodeau. And then this is Jeremy Wright. He's one of the co-authors on the paper that was doing a lot of the genetic analysis. So, as you might recall, we collected tissue samples from several different populations. We collected tissue samples from the peripheral population of spotted gars in the Great Lakes, and we also collected tissue samples from other populations in the sort of core population. We got some from Illinois, some from Louisiana, some from Texas. As I mentioned earlier, we eventually updated this map to cover that Texas population there. We also used the Florida gar as an outgroup. Again, we wanted to use an organism that was somewhat related, but definitely different from our main organism of this study. So we know that all of these populations should be relatively closely related. We use the spotted gar, which is the next closest relative to the spotted gar. Excuse me, we use the Florida gar as the next closest relative to the spotted gar as sort of our outgroup, something to compare everybody else to. So we used 3 mitochondrial genes, the 16S, the CO1, and the CO2. And we broke down these regions, or the populations into particular regions. So we called it the Western Gulf Coast, the Mississippi River Drainage, and the Great Lakes. We did not get any samples from the Eastern Gulf Coast. So, again, looking at these bar graphs, what I want you to pay attention to is the colors. And the colors are going to reflect genetic diversity. So just assume that more colors means more genetic diversity in that particular population. Fewer colors means less genetic diversity in that particular population. So we only had samples of the CO1 gene for all of these particular populations here. So Michigan, Lake Erie, which are both part of the peripheral population, indicated by the letter P here. And then we had Illinois, Louisiana, and Texas as representatives of the core population, as indicated by the letter C. So if we look at the CO1 gene, we can see that Michigan, Lake Erie, and Illinois all had pretty much the same level of genetic diversity for that particular gene. Here we see that we've got 2 variants on that particular gene in Louisiana, and then we have a completely different variation of that particular gene in Texas. And if we look at the CO2 gene, we see something a little bit different as well. Michigan only has 1 sort of consistent variation of that gene throughout that population. Illinois has 2, Louisiana has 3, and then Texas also has 3, but we also see this particular, what we call haplotype, that we don't see anywhere else in our other populations. So if we put all of this together, so all of the genetic diversity information together, and we end up with this sort of series of bar graphs, and then we also have this figure that we can put together. So as you can see in the peripheral population, we have a relatively uniform sort of level of genetic diversity. Again, just looking at these 3 genes. In Illinois, we have 2 different haplotypes, so a little bit more genetic diversity than Michigan, but definitely not as much as what we see in Louisiana, where we have 4 different haplotypes. And in Texas, we actually see a completely different sort of genetic makeup than what we see in Louisiana, Illinois, or Michigan. So Texas is definitely different than the other populations. Here you can see some differences as we go from south moving up north. We see greater genetic diversity in Louisiana in sort of a highly dense area of the core population. As we move further north, we see less genetic diversity as we get into Illinois. And finally, when we get up to the peripheral population, we just see a single haplotype. We see this uniform haplotype here up in Michigan. And of course, Texas is very different here. So Texas seems to be different. So what I want you to think about are those earlier discussions we had about divergence between the Texas population and the rest of the core population. So remember that the Texas populations here are probably not going to be connected to the Louisiana or Mississippi River populations. So Texas has had a fair amount of time to diverge, and I want you to dig into that David and Wright 2017 paper to learn more about those differences there. We also want to revisit our isolation by distance model. If you recall, we talked about this in Chapter 6 and also earlier in the semester with the spotted gar case study. So on our y-axis, we have genetic distance, which is measured by FST, and on the x-axis, we have geographic distance. And as you can see, what the basis of isolation by distance is that you're more closely related to your nearest neighbors than you are to your more distant neighbors. So Michigan is more closely related to Illinois than it is to Texas. So when we compare the genetic diversity across these different populations, we see that you're more likely to be more closely related to somebody that you live closer to than somebody that you live much further away to. And we used our example of those of you from the Bayou region have more relatives here than you would in terms of having relatives up in Alaska or let's say the Pacific Northwest or somewhere very far away. So genetic distance tends to be correlated with geographic distance. So the closer you live to somebody, the higher likelihood that your genetic makeup is going to be more similar as well. So the summary from that particular study is that the spotted gars from peripheral and core populations did exhibit significant genetic variation based on our 3 mitochondrial genes. That Great Lakes population genetic structure might reflect recent colonization. Now I want you to go back and think about the glaciation we talked about and how those populations were probably colonized or founded by Mississippi populations. The other concept that other peripheral populations likely exist, we might be able to apply that to Texas. Although Texas isn't necessarily as extremely of a peripheral population as Michigan is, where Michigan is completely disjunct, Texas has had time to diverge from the rest of the core population, and it wasn't glaciated, and so that might be why we see some of that greater genetic diversity there. So all things to keep in mind when you're thinking about genetic diversity, founder effect, and these sort of colonization effects there. So I want you to keep all of that stuff in mind as you work on case study number 2. And again, email me with any questions that you might have, and we'll kind of transition to our newer lectures moving into the rest of this week. Again, if you've got any questions, please feel free to email me. Thank you, and hope you all are staying safe.